Today I'll be going through Part 2A of Module 2, Chapter 2 of the OCR Biology A-Level Specification. I'll be talking about the ultrastructure of the cell types, specifically eukaryotic cells, from Chapters 2.4 to 2.5 of the A-Level Biology textbook. Part 2B will focus on prokaryotic cells and compare the structures with eukaryotic cells. This video is focused for the OCR Biology specification, but it can be useful for other exam boards. Cells are the basic units that make up everything. Microscopy allows you to see the inner workings of a healthy cell. Scientists have broadly categorized cells into two types, eukaryotes and prokaryotes. Eukaryotes can be plants, animals, or fungi. They can be unicellular or multicellular organisms. They contain membrane-bound organelles in their cytoplasm. Prokaryotes include things like bacteria and archaea. They are single-celled organisms and they have very simple structures contained within their cytoplasms. For your exams, you need to know the ultrastructure of the eukaryotic cells, so I'll go over the organelles and what they do. This part will also include information about animal cells and some information about fungal cells. This is a general overview of what the structure of a typical animal cell looks like. The majority of all eukaryotic cells contain nuclei, with the exception of things like erythrocytes, also known as red blood cells, which don't contain nuclei. The nucleus is double membrane bound by a nuclear envelope. The DNA is contained within the double membrane of the nucleus to protect it. Materials are able to leave and enter through nuclear pores which are found on the nuclear envelope. For example, RNA leaving the nucleus during protein synthesis. The DNA that is found in the nucleus interacts with histones, which form a complex called chromatin. Chromatin coils up to form chromosomes, which you are able to see during cell division. Here you can see the nucleus on the photomicrograph. The nucleolus is a smaller area within the nucleus, and this is the site of ribosome synthesis. The nucleolus is made of RNA, which makes ribosomal RNA that combines with proteins from the nucleolus in order to make ribosomes that are used in protein synthesis. The endoplasmic reticulum are made of membrane-enclosed folded and flattened sacs called cisternae. The rough endoplasmic reticulum has ribosomes bound to its surface. It's the site for protein synthesis and protein transport. This happens on the ribosomes that are on its surface. The smooth endoplasmic reticulum has no ribosomes on its surface, hence why it's called smooth. It synthesizes, processes, and stores lipids and carbohydrates. Here you can see the rough endoplasmic reticulum on the photomicrograph. You can notice the ribosomes on this photomicrograph. Here you can see the smooth endoplasmic reticulum on the photomicrograph. Ribosomes themselves are found throughout the cell, in the cytoplasm or in the RER, and even in some organelles like mitochondria and chloroplasts. These are non-membrane bound organelles. As mentioned earlier, they are a site of protein synthesis. They exist as 70S in prokaryotes and 80S in eukaryotes. Ribosomes are typically composed of two units, which will become more relevant later on when we talk about protein synthesis. The Golgi apparatus, like the endoplasmic reticulum, are made from flattened sacs. They don't contain any ribosomes, and they have vesicles surrounding their edges. The Golgi packages and processes proteins into the vesicles to allow them to be carried out of the cell. They can also produce lysosomes. On the Golgi, the entry face is called the cis face, and the exit face is called the trans face. The cis face faces the nucleus, and the trans face faces away from the nucleus. Here you can see the Golgi apparatus under a microscope. You know it's the Golgi because it has vesicles surrounding it. Now onto the vesicles and lysosomes themselves. Vesicles are membrane-bound carrier sacs, and they store and transport materials inside cells. Lysosomes are a special type of vesicle, with digestive enzymes inside to break down waste materials. They also partake in programmed cell death, also known as apoptosis, and they play a role in the immune response, specifically in phagocytosis to break down engulfed pathogens. Mitochondria are a type of double membrane-bound organelle. They are said to be the powerhouse of the cell, and this is because they're the site of the final stage of cellular respiration. ATP is produced in bulk here. The inner membrane folds to form cristae or cristae, and the interior is full of fluid called the mitochondrial matrix. Mitochondria contain their own ribosomes and even mitochondrial DNA. This supports the symbiotic theory of mitochondria and chloroplasts, as they are thought to have descended from prokaryotes as their own organisms. Here's what the mitochondria looks like on this photomicrograph. You can tell it's a mitochondria because you can see the folds in its cell. These are where the cristae are. The plasma membrane is a kind of partially permeable membrane that's made from a phospholipid bilayer and contains proteins. It controls and regulates the movements of materials in and out of the cells and the organelles. You're going to take a closer look at them later on in the course, but here's a general overview. Flagella and cilia are a type of cell structure that only exist in some cells. They can be found in both prokaryotic and eukaryotic cells, and are extensions protruding from cells. Cilia are hair-like structures that act as sensory organelles or cause materials to move. For example, cilia in the nose detect stimuli, and cilia in the trachea waft mucus down the throat. Flagella are tail-like structures. They allow cell mobility and detect changes in the environment as a sensory function. Both exist in a 9+2 arrangement, where 2 central microtubules are surrounded by 9 pairs of microtubules that slide over each other to cause movement. Cell walls are a type of structure that don't exist in animal cells, but do exist in other types of eukaryotic cells and can be found in prokaryotic cells. Plant cells all have cell walls, which are made of a carbohydrate called cellulose. Cellulose cell walls are permeable, and they allow substances to diffuse in and out of the plant cell. They provide structure, rigidity, and act as a defense mechanism against pathogens, inhibiting their pathway into the plant cell. Fungi can also have cell walls. These are made of chitin. Chloroplasts are a type of double membrane-bound organelle. They're found in stems and leaves and contain chlorophyll, giving them the green color. They are a site of photosynthesis in plants. Their structure contains stacks of grana that are made individually of thylakoids. Each grana is connected by lamellae. The internal fluid of the chloroplast is known as stroma. Chloroplasts also have their own ribosomes and DNA, like mitochondria. This supports the symbiotic theory of chloroplasts previously being descended from prokaryotes. Here you can see the chloroplast on this photomicrograph. You can tell it's a chloroplast because of its shape. Vacuoles are sacs of sap that are lined with a selectively permeable membrane called a tonoplast. They help to maintain turgor in plant cells. They can be permanent or temporary. The cytoskeleton is a really important feature of all eukaryotic cells. They exist all throughout the cytoplasm, and they hold organelles in place and provide cell stability. Also, they provide a way for the cell and its organelles to move. The cytoskeleton is composed of the microfilaments, microtubules, and intermediate fibers. Microfilaments are made from actin and are responsible for cell movement and cell contractions in cytokinesis when the cytoplasm divides. Microtubules are scaffold-like structures. They determine the cell shape and act as tracks for movement of cell organelles. They are what the spindle fibers that are present during cell division and the centrioles are made of. Intermediate fibers provide mechanical strength to cells. They help cells maintain their structure and function. Centrioles are a structure that are made of microtubules. 2 of them associate to form a centrosome that's required to form spindle fibers during cell division. This concludes part 2A of my video on the ultrastructure of cells. The next part will talk about the ultrastructure of prokaryotic cells and compare them to eukaryotic cells. Thank you for watching.